Now let us deal with tapetum. So tapetum has its several features, and one of them is that it digests callose. Yes, in the callose it has some things which helps in digesting. So, and it also has some several features which we are going to deal in this lecture. So the functions of tapetum. Callose, actually it digests callose. Also, it forms a protein wall which has two layers: proteinaceous wall. Protein wall has O, that is the outer layer is known as exine, and I, that is interior, is known as intine. Okay. So I am explaining this formation of proteinaceous wall is in the how the protein wall is presented over tapetum. So let's say this is a body of pollen grain. Like this. Over this there is presence of tapetum which makes look similar to sunflower. It's just a fact, but so this is a cell wall, and inside it there will be a formation of this protein wall like this. This is known as protein wall. So, as you can see here, this is sporangia, the formation of tapetum, and here inside the cell wall there will be a formation of protein wall. Now let's say if here is a pollen grain, then it is known as this. If there is a condition when the pollen grain is presented over the inside protein wall, then this will known as probish body. Probish bodies. And if this pollen grain comes over here outside the protein wall, then this are known as sporopollenin. And when it comes inside here, then this condition is known as, or they are called as Jewish body. And this wall is known as exine wall. Known as exine. Now one more feature of tapetum, that it forms a pollen kit. Pollen kit is nothing but it's a colorful covering over a pollen grain. Yes, it's light colorful pollen grain, and due to the presence of light color, it attracts the insects for the pollination. So I'm writing this information. It is sticky, colorful covering over pollen. So features, it attracts insects for pollination. They are light colors due to presence of pigment carotenoid. And it acts as a shield pigment, a protecting pigment. It is a shield pigment. And what does he do? From whom does the carotenoid, the shield pigment, protects? So it protects from the UV rays. Protects from UV rays. And insect for pollination, in most of, you can see this in most of the entomophilous plants. Now one more information about the tapetum that I am writing here. I think this can be enough. The tapetums are fatty in nature, and they become fatty through the polymerization of carotenoid pigment. Now, the tapetum is also known as most resistant. They are most resistant to the organic material. Because why they are called so? Because they have the capability, they have the ability to withstand in highest temperature too. Yes, they can also, they are known as, they are highest, they are most resistant to organic materials due to they can withstand in the high temperature, can also suffer the strong acid as well as alkali. Now, till now, there is no enzyme ever founded which can degrade the tapetum. No enzyme, no biological uh, thing which can degrade the tapetum. So they are also resistant to the biological degradation. To biological degradation. And due to this, the pollen grains can preserved in the fossil. Yes, they are preserved. Pollen grains are well preserved in fossils. So this is all about the functions of tapetum, and after this we discuss little more about tapetum so that everything about tapetum is clear. Now, as I have already told you before this, that the tapetum consists of proteinaceous wall. So, the protein which is present over the pollen wall, it causes some allergic reactions. So protein which is present over the pollen balls it causes some allergic reaction. Yes, allergic reactions, some respiratory disorders also. Also, one of the example which is caused by the protein which are present over the pollen ball is bronchitis, that is asthma. Also known as asthma. Now I am citing some examples which you can learn by the code word which I am giving, that is caps. Yes. What does it mean? That is C stands for kinopodium. Kinopodium. A stands for amaranthus. And P stands for parthenium. Also known as carrot grass. And S stands for Sergium. Now, this parthenium, which is carrot grass, this came into India as a contaminant of wheat. Yes, this came into India. Came in India as a contaminant. Wheat. Now, second is the protein which are presented over the pollen ball has ability to recognize a correct pollen, a correct compatible stigma. So we can say that it has the capability of recognition. Recognition of compatible stigma. Now, what I mean from compatible stigma? So, there are some stigma formations in plants which cannot digest, which cannot shows the path towards the ovary of a plant, so that the pollen grains cannot reaches to their correct location for the fertilization. Due to the presence of proteins in the pollen ball, they can easily recognize which is a compatible stigma for them, so that they can easily follow the path and reach till the ovary for the fertilization. Now this is all about tapetum, and after this we are going to deal our next topic. Now after tapetum, let us deal about megasporogenesis. Sorry, microsporogenesis. Now what is microsporogenesis? So microsporogenesis is a process of formation of the microspore from the microsporic mother cell through the process of meiosis. So I am writing here process of formation of microspore from microsporic mother cell through meiosis. Now, as I have told you in previous lectures, is the meiosis consists, it has two phases, that is meiosis 1 and meiosis 2. And the meiosis is also a reductional division. So before dealing about the types of microsporogenesis, I am going to deal about two types of karyokinesis. So, two types of cytokinesis. That is successive and simultaneous. So, the successive type of karyokinesis, the process of karyokinesis occurs, you can see, in monocots. And the simultaneous occurs in dicots. Yes, successive type of karyokinesis you can see in monocots, and the simultaneous one occurs in dicots. Now let's say this is a cell. Let's say this is a microsporic mother cell. That is MMC, and this undergo division through the first phase of meiosis, that is meiosis 1, goes to the first phase, that is meiosis 1. Now here karyokinesis is present, cytokinesis is also present. Now, as the meiosis is a reductional division, what happens here? This cell will show growth, and the one wall will form to and divides this whole cell into two parts. So this results in the ultimate division of this microsporic mother cell into two cells, and this plate is known as cell plate, which divides the cell into two parts. Now, after this cell undergo the second phase of meiosis, that is meiosis 2, and now again there will be a formation of one more cell plate which forms vertically, and now there are four cells. Due to in the first phase, this cell divides into two due to the formation of cell plates. It is known as dyad. Yes, dyad type of cell. And this here also the presence of karyokinesis and cytokinesis too. This is known as dyad, and there is the formation of four cells, so it is known as microsporic tetrad. Microspore tetrad, also known as isoBitetrad. Now we talk about dicots. Let's say this is a cell, the microsporic mother cell. It undergoes the first phase of meiosis, that is meiosis 1, and it divides cell into two. But in the case of simultaneous type of cytokinesis for dicots, it divides into two cells, but there will be no formation of cell plate as you have seen in monocots. So here is only present of karyokinesis, not cytokinesis, when it undergoes meiosis 1. Now after this, when it undergoes the second phase, that is meiosis 2, it takes place, there will be a formation of cytokinesis, and it shows shape like this, tetrahedral type of shape.